All right, so today we have the esteemed pleasure of having Dr. Lorin Crawford. He's a senior researcher at Microsoft Research New England. He also holds the position as the RGSS Assistant Professor of Biostatistics at Brown University in Providence, Rhode Island. His interests involve the development of novel and efficient computational methods to address complex problems in statistical genetics, cancer pharmacology, and radiomics, otherwise known as cancer imaging. His most recent work has earned him a place in the Forbes 30 Under 30. I passed that a long time ago. 100 most influential African Americans list, and recognition as an Alfred P. Sloan Research Fellow, and also a Packard Fellowship for Science and Engineering. I was also a Packard Fellow. And I think you were also on, was it the CBS Good Morning America?
Yeah, that happened recently.
Yeah, I think that's what he's most famous for, was being on Good Morning America. In 2017, he received his PhD from the Department of Statistical Science at Duke University. That's in Durham, North Carolina. And he received his bachelor's degree in mathematics from Clark Atlanta University, where he had a 4.0 GPA and was the class valedictorian. So today he's going to share with us his journey through academia and industry, as well as provide his view on the critical importance of interdisciplinary collaboration in the emerging world of data science. I'll note that when Lorin once said that, quote, "The honors program at Clark Atlanta gave us family to step into. If we have problems, we could turn to one another. It helped us find out who we are and what we want to be. If we were lost, we had people we could trust that would steer us in the right direction." So we thank you, Lorin, for continuing this legacy and providing us data insights to help steer us in the right direction.
Awesome. All right. Thanks so much for the introduction. Wow, that was a lot. It's a pleasure to be here. I wish I could go. I wish I could be there in person because I really miss Atlanta. So thanks everyone for joining and tuning in. Today I want to talk about this idea of sitting at the intersection and my kind of view on the world of data science. Some of this might have already been said before, but I'll kind of go through my journey a little bit. So I like to show this slide because I saw a colleague of mine do the same thing, where on the x-axis you have years, and on the y-axis you have the cost it used to cost to afford to sequence a genome. And for those who don't know, I'm a computational biologist, and so this kind of goes right in line with my idea of looking at my life from a genomics perspective. And what my colleague did was she overlaid her own timeline on this plot. And so you can see the cost kind of going down with respect to Moore's law. And she would say something like, in 2003, I graduated college, 2000 such, my first child was born, and so on and so forth. So I thought I'd kind of do the same thing and kind of give people a relative timeline of my own life. So in 2003, I didn't graduate college, I graduated from 6th grade. In 2009, I graduated from high school, and I moved from Los Angeles, California to Atlanta, Georgia to attend Clark. There I studied math, and I graduated in 2013. And then I moved on to Duke to study statistics. In 2017 is when I graduated. I kind of became an adult that year. I graduated grad school, got married, and started my first faculty position at Brown. And I just recently moved to MSR, Microsoft Research, last year. And so, yeah, Clark was an amazing time in my life. I actually think that I wouldn't be here without my time at Clark Atlanta University. It was there where I first was introduced to this idea of math and biology kind of colliding in Dr. Sandra Rucker's class, where we had to study using differential equations, the rate of mutational change for HIV. And I thought that was like a really cool application of using mathematics to actually solve these like real world problems. So I graduated Clark, like I just mentioned, as valedictorian. There in the corner with my roommate, Tam. And from there, I actually moved on to Duke, where I studied statistics. There I was motivated to study statistics from Dr. Abebe at Clark Atlanta, who was a Sachs professor, as well as my mentor and advisor while at Clark. And Duke was actually an amazing transformative time in my life. It's where I really got to see this idea of not just colliding two worlds together, but try to figure out how to collide these other as many possible worlds together as I could. And I had a lot of support to be as creative as I could while I was at Duke. So there I was in statistics, but I was co-advised by someone in pharmacology, cancer biology, Chris Wood, up on the bottom right-hand side. And so I kind of sat between doing computational work and sitting in a wet lab, actually. I didn't do any experiments. I've never pipetted in my life, but kind of just being around this world of molecular biology and kind of teaching myself this vernacular that was really foreign to me was amazing. So I remember my first summer there, these words like apoptosis and all these things that in biology that I hadn't even picked up since high school were, flying over my head until one day, I was able to piece pieces of the puzzle together. And so I spent 3 out of my 4 years in the pharmacology cancer biology department, kind of doing computational methods and trying to understand things like, can you build statistical models to understand phenomena like resistance to cancer therapeutics? While I was also there, I also spent a lot of time in Columbia, in New York. Cheyenne, my advisor in the top right, was really supportive of me flying around and trying to meet as many people as possible. And so I met this guy, Raul Rabadon, who studied topology and used topological data analysis to understand the evolution of cancer and individual cells. And so he was actually really nice enough to let me come and fly and hang out in New York like 10 days at a time, my 3rd and 4th year. And so I kind of got to learn TDA. And I was able then to piece statistics and this cancer biology, genomics, genetics with this topological data analysis to kind of form this like really weird research plan. And when I was graduating from Duke, my advisor kind of had this crazy idea for me to apply straight for faculty positions and not apply for postdocs. And so I, shot in the dark. I kind of sent my applications around to different universities. Took a visit to Brown, absolutely loved it. They were willing to hire this like 26-year-old kid that had this really weird idea of mixing these 3 disparate fields together. And they took a chance on me. And so I became a faculty member in the Department of Biostatistics in the School of Public Health, as a core faculty member in the Center for Computational Molecular Biology and the Carney Institute for Brain Sciences. I have a lab of about 8 or so people. It's been a lot of fun. Brown is a really cool place, and I could talk to anyone who's interested about opportunities, and I would love to get into that later. It's a really flat university, and so if you're looking to be creative, and especially you talk about the space of data science where you can kind of mix and match fields and disciplines to create your own lane, Brown is an amazing place to do that. And Brown's strength is collaboration, so I felt right at home when I stepped on campus there. So I've been at Brown now. This is my 5th year. I started at Microsoft Research recently. Some good things have happened along the way. The Forbes thing, the Sloan Packard Foundation has been absolutely amazing. This idea of being en route with like people like Steph Curry and Nipsey Hussle and Stacey Abrams is also kind of wild. But it's been a lot of fun thus far. What I really loved about my journey through academia is this idea of building interdisciplinary teams and doing this kind of team science approach to stuff. So at Brown, I have my own research group of, like I was mentioning, about 7 or 8 PhD students, a team of undergrads and master's students, and everyone works collaboratively, collectively together. What I love about my time at Brown also is that the people that you're seeing, at least on this slide, span many different departments. And so you have a few who are in applied math, a few who are in biostatistics, a few who are in computational biology, as well as a few people who are in physics. And so it's been a lot of fun thinking about how to create and figure out where we can, again, meet at this like intersection of disciplines to do like revolutionary science. My time at Microsoft has also been equally as enriching. The Microsoft Research Biomedical Machine Learning team is also this idea of interdisciplinary science. Everyone you see on this slide here are coming from areas like bioengineering to thinking about protein structures and healthcare, and I have this weird thing, and so it's really cool to also, again, Microsoft thinking about this idea of a flat place. Microsoft is also a very flat company where people can come in and create, and it's welcome to bring new ideas. And so I've had an amazing time there. Again, also would love to talk about opportunities at Microsoft and Microsoft Research. So like, who am I and like, why am I here? So, a recurring research theme for me is to take modern computational approaches and develop theory that enable their interpretations to be related back to the basic genomic principles. So what I say about that is that I'm a statistician by training, but I don't just build really fancy hammers and then go look for nails to hit out in the world of data science. I really care about biological questions and biological problems. Then I try to build really specific hammers for that particular nail, right? And so a big part of my research program is this idea of dissecting phenotypic variation, right? So you can think about this idea here. We have a panel of mice phenotypes, anything from like body mass index to height to behavioral traits. And what you could do is you could think about how different genetic components or environmental components drive the variation of these traits across different populations, right? And you think about how the architecture of those traits might be different if I'm looking at people of, say, European ancestry versus someone of African ancestry, right? Or you think about someone in one environment versus a different environment. You compare how these architectures of traits might differ across those different axes, right? And so I think about phenotypic variation as like an entire pie, right? Like we all have traits in our and phenotypes that, in our bodies, in our cells. And you could think about something like height or something being an entire pie. You can break that pie down into different components. And so part of your phenotypic variation is based on genetic traits or genetic factors, as well as environmental factors, right? Now, my group, we think about taking that genetic piece and then breaking that down even further, okay? So the genotypic variation can be dissected into something like additive. So you can think about how one gene plus the effect of another gene gives rise to a trait. But we also really care about this idea of nonlinear variation. So the effect of one gene multiplied by the effect of another gene, right? This like nonlinear component, right? And we consider these nonlinear components to be epistasis, but you can also think about a nonlinear component being how one gene interacts with another environmental component, right? And how that interplay might change depending on, again, the type of person or type of populations I'm studying, right? So the types of data that we work with are actually really cool. They're really high dimensional data sets, right? So we have some like continuous vector of traits. You could think about this as anything from like crop yield for like plants to height in human beings. And we study this based on genetic factors. So we have like these high dimensional matrices that we get. And so these, you can think about this as, in individuals or in observations, we have P genetic features. These can be genes, these can be mutations or SNPs, right? And what we want to do is we want to relate these individual vectors of these little individual genetic features or genetic markers to the variation of that trait, right? So which of these features are most important in explaining that Y variable, right? And there are many cool ways to do this. Linear models are typically how people think about this. And so, I have again, my high dimensional X matrix. These beta vectors here are really important because each of these betas you can think about this as a weight, right? Like how much does one genetic feature influence the outcome of my trait of interest, right? Now, this is a linear model. So what we're only really looking at here is this idea of gene A plus gene B, right, in a linear fashion. Now, my group thinks about how to go beyond this linear trait mapping paradigm. And we look at using machine learning and black box statistical models to do so, right? So machine learning are typically well suited for prediction-based tasks, right? You've seen this probably in anything, even from predicting which ads someone might like or interact with, to which treatment strategy might be most suited for this given patient, right? So when I say black box, what I basically mean is that the statistical machinery that's used to make these predictions, there's not a lot of theory or understanding of what's actually going on inside these models, right? I don't necessarily know how my machinery is upweighting or downweighting my features in order to make this really nice prediction, right? So what I effectively mean is I have some input that goes into my algorithm, outspits some really nice prediction, but you really have no idea what's happening inside this black box, right? So my group's research goal is to provide interpretable ways to summarize the importance of my input features in the context of this algorithm, right? Now, again, I consider myself to be a computational biologist, and so I think about this as being genetic features and my output as being phenotypes or traits of interest, okay? So, the crux of this is this idea that linear models, which are conventionally used in the field, only see a small piece of that pie that I was mentioning earlier, right? They can only model this additive portion, right? Whereas black box machine learning methods can model, the entire spectrum, right? Now, the difference between the left and the right-hand side is I have interpretability on the left, right? I don't have this black box idea. I have these weights that I can do ranking of my genetic features with, and I don't necessarily get that on the right. And so my group thinks about innovative ways to do the right-hand modeling while still getting the interpretability that we would get on the left. Okay. So, the one example of this idea that like you can use real domain knowledge in order to inform what your model should or shouldn't be learning, right? This is, again, meaning at the intersection. I can use my knowledge of biology to inform what I should be doing on a statistical or computational level, right? So let's just take a really basic example. In biology, we have this nice hierarchical nature to enrichment studies. So on the x-axis, you'll see chromosomes. This is based on type 1 diabetes in human beings. We have 22 chromosomes. The y-axis gives you a importance or probability score. And basically a spike in a region means that there's an enrichment for that given trait at that given genomic position, okay? So a large spike means that there's a huge enrichment, right? So you can see in what we call the HLA region on chromosome 6, there's that really nice spike. Type 1 diabetes is an autoimmune disease, so you would expect to see that there. Each dot here is a SNP. It's a mutation marker for that we know is located in that given position on that given genomic location. What's really cool about biology and our knowledge about biology is we can actually annotate each of these mutational markers for a given gene that we know encodes that region, right? So each of these genomic positions can then be grouped together in order to say that these groups of mutations are related to this given gene of interest, right? So why that's really cool is that we can actually use this structure to inform what a machine learning method might look like, right? Let me give you like a really basic high-level example of how this might look. So, I have this kind of predefined gene list, right? Where I kind of know that these genes lie on this chromosome, right? And I know that these genes start and end in terms of their coding regions in this given window. And then I know which SNPs, right, belong in that given window group, right? So I can use that information to inform what my architecture of my model might look like, right? So let's just take neural networks, for instance, right? Now, stay with me. What's really cool about this, you can actually visually see this. If I flip this graph on its axis, this grouping, this partial connected grouping, looks like what we call a neural network, right? And so again, I've used like my domain knowledge to define what I should be using for my model space, right? What's really nice about this is it gives a nice interpretability across the different layers of my model. Right? So now my input layer has some kind of meaning of these are individual SNPs. My hidden layer means that each only SNPs that are encoded for the same gene get connected together in the second layer. That gives my second layer this idea of being a gene or SNP set layer, right? And then my output, I model my phenotype, right? And so I've given my model interpretability all the way through, but again, it's this idea of being motivated by what I've learned in biology, right? And so this is really cool. We kind of wrote a whole paper on this and we made it a little bit more general, right? Anytime I have some groupings that I know to be true based on biological annotations, I can build a nice machine learning model to represent that, right? And then I can be really mathy about it, right? So once I have this kind of nice hierarchical model, I can build a full probabilistic specification of how this model should be fit and then what I can actually learn from it, right? And so here I went to Duke, so I'm a little bit Bayesian in how I think about things, but we can be very probabilistic in saying that once I kind of know this partial connected structure, I can learn how important any of these thetas are, which means I can learn how important any of my SNPs are. And I can also learn how important any of these Ws are, and that'll tell me how important any of my genes are. And then I can go one step further and then figure out, okay, which SNPs are most important in the context of my traits, so which mutations are most important, but also which genes are most important. And if I know that, then I can say, okay, how do I now build drugs or something like that in order to think about treatment strategies now that I know the variation of what's being dysregulated across this population of people. All right, so let me just give you a really quick example of this. Here are a few cholesterol traits in 2 different consortiums. Again, we have these what we call Manhattan plots, right? 22 positions on my, or 22 chromosomes around my genome. Again, an enrichment score, these what we call inclusion probabilities, which means the probability that that given mutation is actually significant with respect to my given output. A spike or a larger close to 1, again, means that is more significant. What you can see is we're able now to do this really cool trait mapping where in these two populations of individuals, I can say, okay, which mutations are most important? And I can also map these mutations to, again, in this legend here, which genes are most important with respect to these given patients or individuals. So that's one way that that's one question that my group actually thinks quite a bit about, like is how do we take these biologically informed stances on how we build interpretability into our models. Now, there's one other space that we've been really creative in, and this goes back to my journey through academia and industry of incorporating like topological data analysis, which might sound a little far-fetched and a little out there. But a part of my goal in my time with the Packard Foundation and my time at Microsoft Research and at Brown has been thinking about how to take these really complex mathematical ideas that seem very theoretical and very far out there, and how to make them more tangible for for things that can be used in algorithms and everyday analyses. And so part of a motivating question for me recently has been, what if I don't have access to genetic data? And there are a few areas where this could be super important. So one area is in animals, right? So here you have a collection of bird beaks on the left-hand side. On the right-hand side, you have collections of fossils. What if I can't take or access genomic sequencing for these particular model organisms? However, I do have access to other parts of their data, right? Or other parts of their organism. Another example of this could be in radiomics or clinical imaging. A lot of us know that, for example, brain cancer, in order to get genomic sequencing from individuals, is quite invasive for patients, right? And also the disease is very rapid in its and people typically decline quite quickly after diagnosis, right? And so, one other example here is, if I can't actually get genomic sequencing from certain individuals, can I actually look at a brain scan or a brain tumor and then relate the morphology of that tumor to the underlying biological processes that might have created it, right? So not ever cutting into the individual, just looking at a brain scan and say, I recognize this particular feature in this tumor. This looks like it's connected to a certain mutation. I should give this person drug X, or I shouldn't give this patient drug Y, right? So connecting this idea of shape variation to like biological phenomena, right? So if I look at a shape, do I actually know what drug agent I should use to block a certain activation in a given signaling pathway, for instance, in this individual? So, and a very last example of this, and something that's been quite important recently, is where it's too expensive to get data, right? So one example of this is in proteins, right? A big example of, or a big application area is understanding how proteins fold and change over time, and that might tell you their biological function. This is used for, again, drug discovery and things like that. And so one question here as well is, well, maybe I don't necessarily have enough sequencing money to sequence over fine stops along this trajectory. Instead, what I want to do is watch how a protein might move over a given trajectory, and then be able to relate that movement to, again, the biological processes that are causing or associated with that given movement, right? So my group has been thinking a lot about this, and here we use topological data analysis actually to do this. And so we created these pipelines. Here's an example of one, what we call SINATRA, to do these kind of tasks, right? Again, this idea of meeting at the intersection. And I love this idea of SINATRA because it has a play on words for me. One, it stands for some acronym that I won't bore you with. But on the other side of the meaning, it also has this idea of, my lab does many different things: statistics, biology, physics, applied math. And this SINATRA is like the one project that we have that kind of takes these disparate sounds and mixes them all together as one coherent project. And so SINATRA is an algorithm where you give it structures of data. So let's say you give it protein structures of a wild type and protein structures from a mutant population. SINATRA then takes these structures and creates what we call topological summary statistics over them. So we take these structures, we run these sweeps over them, and what these sweeps do is we count what we call Euler characteristics at every stop along this trajectory, and we create these curves, as you're seeing in panel C. And what these curves represent are the geometry and the topology of that given structure, right? So we take shape, we turn it into these curves. What we do with these curves is we model them. We try to understand which pieces of these curves are different for the wild types versus the mutants. And so on the bottom here, you can see these red areas. The red parts of those curves are telling you which part of those curves are most different or best describe the variation between a wild type and a mutant. What's cool about our transformation from shape to curve is that it's invertible. So once I go from shape to curve, I can actually go back from curve to shape. And why that's important is because it now allows us to highlight on the actual structure what, in terms of like actual 3D structure, what's most different between those two groups, right? So here's a basic example of this. These are like structural, these are two different chains from HIV-1 protease. And you can see these red regions are the regions that are most enriched for being the difference in terms of how these proteins move over time between if I was looking at chain A versus chain B, right? And again, this might inform drug applications and things like that. So it's really cool. And again, this is the idea of mixing statistics, biology, physics, and a little bit of topology. And so that's like a little bit of overview of things that we're doing. And I know I'm quickly running out of time here, but this is the idea of us kind of meeting at this like intersection, right? And so I kind of want to leave here, and I could probably spend an hour on each of these, but kind of giving everyone these things and key things that I've learned thus far as hopefully takeaways of like my journey, and I just kind of want to pay some of this advice forward. For people who are listening, you know, especially students, it's really important, I think, to create a network of pseudo advisors outside of your immediate workspace. My advisor, Cheyenne, was really big on me having people to depend on outside of him, right? This idea of I could go to someone and ask them for advice, and even though they may not have been directly involved with my career trajectory, you know, they were invested in it, right? And so anytime I needed something, letters of recommendation, ideas on where to do internships, where to apply for, like I had other people outside of my advisor to lean on. And that has helped me throughout my career, especially looking for people who are one arm's length away from me, right? And that's been huge. I always tell my lab this, and I want to keep stressing this, particularly during the pandemic, when work-life balance has become even quite more difficult for a lot of people. You know, take the time to celebrate all victories, especially the quote-unquote little ones. And I say quote-unquote because they may seem little to you, but they're probably not little in the abstract, in the whole, in the abstract, right? So, you know, take the time to celebrate little things. If you improve on one thing, if you learn one thing, for graduate students, if you finish a draft for something, even though it doesn't get published, like take the time to celebrate the little victories along the way, and that'll really help you be able to endure these ups and downs throughout research and academia or even industry as you navigate new spaces and new terrains. This is a huge thing that my co-advisor Chris taught me. Is this idea of thinking about where your field is going to be in 10 years, and then figure out how to position yourself for those new horizons now. And I used to spend a lot of time thinking about this, of like, where is technology going, right? Going back to my genomic timeline, right? This idea that sequencing continues to drop. So, you know, what am I thinking about now, right? If sequencing is getting cheaper, that means people are going to be able to do more, right? And what does more mean? And how do I set my lab up to be there right when more happens? Right. And so, you know, take time to really think about those strategic moves. Again, this idea of like a chess piece, right? These strategic moves as you go throughout your career, right? And, you know, think about the immediate, but also think a little bit further down the line. The second last thing I like to say is remember to work on your soft skills as much as your technical ones. Writing, giving talks, interacting with colleagues, right? Working on teams, you know, being able to also improve in those intangible ways as well as the ones that are more tangible. That's also been a huge thing for me in my career thus far. And the last thing I would say is invest in the time to learn multiple scientific languages. Just like in business, where speaking multiple languages is seen as a plus, I think the way that data science is going and the way that lines are continually being blurred, and even in some of the projects that I just showed today, I think this idea of being able to move in between communities is going to be huge for people. I just think that's the space, that is the direction that data science is going. It's not so much about what you're majoring in anymore, or what your degree's in, right? It probably never mattered, but I think now more than ever, it's about skill sets and being able to communicate to different types and different people with different expertise. And so, definitely take the time to learn different things. Don't just work in silos. I don't think that's the way that data science, I just don't think working alone is the way that people in data science are successful. I think that, this interdisciplinary collaborative spirit is what's going to continue to push this field forward. So for that, I think I'm going to end actually on time, which is amazing. This has been a huge effort from a ton of people, as you can see, across many different places, career stages, and even different industries. And so it's been really fun working for me in these spaces and having the support to kind of do that, whether it be at Brown and now at MSR. People have given us money, which I've been really thankful for. Here are some relevant references for anyone who's interested. And with that, I'd just like to say thank you so much for listening. I'm happy to take any questions, happy to talk about Brown and all the cool things that are happening on that front in terms of new programs and new initiatives, as well as things that are happening at Microsoft. I think that Microsoft's in a very cool place right now, particularly in the health and biological spaces, and thinking about how to be super innovative there. So I'm really happy to talk about opportunities there. But yeah, thank you so much.
Thank you so much. That was amazing. Very insightful. Love the tips. So thank you so much. We're going to give you a little breather here. And I think Bettina did a poll. Bettina, were there any poll results that you saw there?
Yes, we have some poll results. And while I'm presenting them, please go ahead and put your questions to Dr. Crawford in the Q&A section. You can submit questions about his life, career path, anything about advice. This is a great professional development session. We want the HBCU students in the symposium to ask as many questions as possible. So looking at our poll, we want to know, how did you hear about our symposium? And the biggest response was from their AUC institutions. So from Morehouse School of Medicine, Spelman, and Clark Atlanta, which is great because we hope that we got the message out to all of our member institutions. There were also a number of people who heard word of mouth and through the DSI, which is our Data Science Initiative newsletter, which we'll drop a link for you to subscribe to if you're not already subscribed. At today's symposium, I am most interested in, we had 24% interested in getting an internship, another 24%, oh, that's on there twice. So we'll count that one as mentorship. And then 7 or 21% interested in getting a full-time job. And then the largest percentage, 54, was networking with other HBCU students interested in data science. So I would advise you to, while in the symposium, go to the People tab and look at who's here and click the connect button, because you can match with them that way. And also sign up for any of our networking and expo sessions. Where do you currently live? So we have 62% living here in Georgia, and then 37, almost 38% that are not in Georgia. And this time, no international, but we'll work on that. By next year, that'll change. If you are a student, what level are you? So we have 21% are sophomores, 5% are juniors, 21% are seniors, 26% are master's students or PhD students. So we've got a number of upper level students here. So definitely we want you in the networking. Definitely submit your resume, no matter which level you are, to our resume book, because that will be going out to our sponsors and exhibitors. And then what is your status? Because we have a mix of people who are here in the crowd. We have 71% are currently enrolled students. So thank you so much for taking time out of your busy semester to come in and join us here. We have 9.5% that are graduated working professionals. Those are our sponsors. Those are our exhibitors. Go check out SAP, Coca-Cola, Claremont Graduate School, Columbia. We have all sorts of people that are here in the expo. Please join and talk with them. And then we have 19% who are faculty and staff, and that includes all of our AUC institutions, but also a number of representatives from different HBCUs such as Morgan State, FAMU, Bowie State, Jackson State, Howard University. We've got a great collection of people here. So thank you all so much for joining and participating in the poll. And I will, we can move on to the Q&A session. So please drop your questions in the Q&A. Thank you.
Thank you so much for that poll report out. And Laura, on behalf of Dr. Rucker, who's the chair of the Department of Mathematical Sciences, she said, great presentation, Lorin. We are so proud of you.
Oh, that's awesome. I miss Dr. Rucker. I hope they're all doing well.
Yeah, they're blown away. Somebody kept saying, sending me emails. I won't say who it is. How wonderful you are. I'm like, put the question in the chat. So we'll start with some questions.
Yeah.
Somebody has a question. Any suggestions on how to pivot from a career in research and development into data science? Yeah,
that's a really good question. That's a really good question. My advice probably would depend on what career stage that individual is in, but I will say uniformly, there are a lot of free forums that allow you to learn like coding in Python and things like that on the side. Those are huge things to take advantage of. The other thing is most companies now also have allotted time for people to learn new skill sets, right? So a lot of companies also give you there's some programs that allow you to have, 20% of your time throughout the week to spend learning, taking the course, working outside of like your immediate responsibilities. And I invite people to really like look into those as well. For more tangible things, if you're looking to get back into like more like academic research, shoot emails to people. Like, if you're in a certain city and there's a neighboring university or something, shoot an email to someone who might be doing computer science or somebody who might be a little bit more even industry facing at these universities. Most people are super nice and most people want to get back and help people. I think the barrier here is on the other side, people are afraid to send those emails because they don't want to be bothersome to people. But most people want to help. Most people want to learn and expand. And what you don't recognize is that from the professor's point of view, it also helps them expand their portfolio as well as working with new people and that kind of thing. And so a lot of times it's mutually beneficial. So yeah, shoot the email and see. But that would be my tangible way of going about it and doing it.
Now, Lorin, I know you played basketball back in the day. So I think to summarize, is it shoot the shot? Yeah,
shoot your shot. Absolutely. A lot of my career has been like shooting your shot.
You never know.
Yeah, to me, I remember talking to Dr. Bebe when I was applying to grad. Like Duke was my long shot school, and it was a school that got back to me first. And so you never know what could happen. The worst that someone could say is no, and if that happens, then you're still in the same position you were an hour ago. So yeah,
that works. That's good. So someone asked, they say, I love the way that you are being such a translational scientist who applies interdisciplinary approaches. What advice would you give to students who are typically, and faculty, I'm going to add that in there, who may be confined to the research interest of their mentors or their specialization, which has its advantages, but it also has its drawbacks?
Yeah.
What's your advice there?
Yeah. So, right. So there's a lot of positives, particularly for faculty, for working your specialization, right? Because it's easier to get grants. It's easier to publish, like your community knows you. There's a lot of things that go wrong with like reputation and recognizability and things like that. What I like to do on the faculty level is it really helps to have collaborators where we can kind of talk, and then we have like a student maybe working with us that can kind of bridge our two interests. I know nothing about protein. I knew nothing about proteins 8 months ago. So there's another woman at Brown who's a computational chemistry. We can kind of talk about statistics and we linked that way. And that was really, really helpful. And so finding a collaborator where you can both teach each other through some projects is an easy way, I think, for faculty to really do that, especially if you have a student that's also involved. For students, I got into biology. Biology is really interesting because it's not like math or other areas of STEM where you need to learn step 1 before you can move to step 2. And if you don't know step 1 and step 2, there's no way you're moving to step 3. Like that's just like math is a very much of a stepping stone. Biology is interesting. It's like it's a disparate. It's like you think about a puzzle. It's a lot of disparate puzzle pieces. And what you have to do to be good at biology, or to become comfortable in biology, is spend time reading. And what that does is you start seeing terms that you see over and over and over again, and that allows you to put these pieces of the puzzle together. You might not have a picture of the whole thing. Like that's hard, but you might have a little neighborhood of like, I kind of understand this concept, and you slowly build out that way. And there are a lot of fields that are like that, where if you spend enough time dedicating yourself to really, going to talks, hearing like what people are interested in over and over and over again, you'll start to see connections between what you're really good at and what they find to be important. And then you can start slowly moving yourself over into becoming more interdisciplinary that way. And that was really helpful for me when I first started as a grad student.
Yeah, I when I was a grad student, I went to UConn, and I was math, but I wanted to go into mathematical biology because back a couple of decades ago, that was the up and coming thing. And so I didn't really have any biology training, but I could read.
Yeah.
I could read and I could learn. And so, yeah, I love all things cell biology, but I did find that knowing the Latin roots helped a lot, because
it does.
Biology words is like German, right? It was linked together, all these different Latin roots together, and voila, you have like a 30 letter word.
I have friends that I work with now that used to make fun of me to this day about when I was in grad school. Like my Google tabs would be crazy of like just words that I was like, I don't understand like what this means, and I'm just gonna have to Google.
Yeah. Where's my Latin dictionary?
Yeah.
Totally good. So someone asked there, do you recommend doing a postdoc if you are interested in going to biotech? Since you did a postdoc, actually,
I didn't do it.
You did do the postdoc. So you did the PhD at Duke, and then that was it. I actually did the PhD at UConn and then did a postdoc at Duke. It's kind of maybe a step behind you there. So I guess it's maybe, what are the pros and cons of postdoc? And okay, let me read it. I'm going to put a not in there. Since you did not do a postdoc, what skills separated you from others that went into similar positions that you've already held? Let me read it that way. So if you didn't have the postdoc, what did you have that kind of circumvented perhaps that quiet expectation?
Yeah. Okay. So this is a really good question, and something I talked about to my lab, especially because I have a lot of people graduating this year. We've had a lot of conversations about this. I think what allowed me to go from my PhD to my faculty position was this idea that when I was on my PhD, my advisor was very hands-off, like very. And so I was able to create my own research program with his kind of high level support versus his very like hands-on day-to-day support. And so I think that what separates a faculty member from other people is that you're able to see research direction, and you know how to go in that direction. You also know there's like this intangible skill of where you know something's not working and you know how to pivot. You know how to like, there's this way of like reading the landscape that I think a faculty member has. And I think this is universal across all institutions. Like you're able to see where research is going, you know when to pull the plug, you know when to full steam ahead. I got that as a graduate student because my advisor is very hands-off. Our relationship was like, I wrote a paper and I would come to him and say, here's my idea. And he would be like, that's bad. Or like, you're not thinking big enough. Or have you read this person's work? You should take that in consideration and maybe think about other ways to pitch this. Like he was about that kind of training, not necessarily about this day to day. So when I went on the faculty market, I knew what I wanted my lab to do. And I had a huge research direction of like, I know what I want to do. I know what my lab's going to be about. Here's my elevator pitch. I think the positive part about the postdoc is if you don't necessarily have that, the postdoc gives you time to figure out what that might be. And what the postdoc also allows you to do is allow you to learn new things. So if you're thinking about this question about learning a new skill set, a postdoc is a great place to actually do that. If you want to add something else to your repertoire, like I think the postdoc is a great time to explore new areas. I didn't necessarily have the time to do that. And people always ask me, like, would I have done the postdoc? Like, I think from my family perspective, it was great because we came out of grad school at the same time, and my wife's a lawyer, so she wanted to start working, and so I think it worked out for us. But I think, when we were, the positive part about doing a postdoc is it really gives you that extra time to really think about some of these things. And I didn't necessarily have that. But for my students, I'm always telling them, like, if you're not exactly sure of what you want to do, because when that tenure clock starts, it starts, and it doesn't stop. And so, having that extra time to do things, I think, is where the postdoc is great.
Great. Yeah, that's how I used mine. I wanted to pivot deeper into biology. So that's exactly how I used it. And, I had a couple more kids, but, it's counting. So I was talking with a student the other day. So I'm teaching a course, Explorations in Data Science. It's very project team. We actually work in class as a group. Justin De La Cruz from the library is joining up with us. We're looking at U.S. Census data. So I was chatting with the student after class who's here. And she was saying that, teamwork, sometimes that can be challenging. And you said that working alone really doesn't lead to success, that it really is this collaborative work, this teamwork, this project team work that really fuels innovation and fuels success. Well, what are your tips for having or contributing to or existing in a functioning, let's say, project team? And what do you do, especially when you're kind of like, how do I fit into this team? What's your approach and that's
a really good question. So there are a few, every collaboration for me is different, but when I first start any new collaboration, one of them is trying to understand what the large goal is. And almost having this like trickle down tree effect, like here's this large like North Star that we're all heading to, and then trying to figure out how each of us might contribute to that North Star. And kind of not necessarily laying out duties, but laying out necessarily like who is going to bring what from like a high level, like to us contributing to this North Star. So a lot of my projects are with people who do wet lab experimentation. And so for us, it's like, it's very clear of the wet lab side is going to be this, and then we're going to handle the computational side. For my computational collaborators, there's a little bit more nuances of, let's kind of lay out right away that this half will be your thing, this half will be our thing, and really trying to, I think a lot of people approach the idea of talking about roles too late in a project, and then everything kind of becomes muddied and you're not really sure what your contribution is. Having those tough conversations up front is much easier. And in academia, I think we all kind of know about this, like authorship conversations, who's going to be positioned where. Having all those conversations up front before you get too far in the weeds it's much easier, because then people don't get lost. You kind of know, like, this is what I'm doing, and then if you don't follow through, like, that was on you because you had your thing. But I think it's much easier, and it's been like that from all of my grad students as well. It's like, this is what you're going to contribute to this, and let's kind of clearly define that before there's any kind of exchanging of data, before there's any kind of let's hit the ground running. Let's kind of have all that planning out right away. And so, communication in teams is huge. And it's much easier to have that communication first before doing anything else. That's been my approach.
Well said. Yeah. Good planning and good communication. Very well said. So someone says, could you give some advice to HBCU students who may be perhaps reluctant to pursue academic opportunities in the majority institution space in, let's say, Rhode Island, in Wyoming, in, Montana, I'm just kind of naming some places randomly, don't know why. So could you give some advice to the students who are, to think kind of outside and kind of go outside of their comfort zone?
Yeah. So first I want to say that my time at Clark, I think, gave me the intangible skills to do well at one of those larger schools. Clark taught me how to hustle for stuff that I, like these intangibles of, like, if I want something, I have to figure out how many signatures I need to get for this thing to happen. And that's something as simple when I was at school, like as simple as MATLAB. Like, if you want to figure it out, you need to figure out who around can help you get that. Clark taught me how to hustle, and it taught me how to persevere. those two things are like super important for people, especially in academia, but people in grad school in general. And that perseverance part is huge because everyone hits a wall, and the people that succeed are the people who are able to persevere. I might be biased, but I think HBUs just teach you how to do that. When I got to Duke, and this is important for me to always tell people, I struggled my first year, like struggled. I failed my first year qualifying exam. Like I thought I was gonna have to drop out. Like I went through it because it's almost like, and again, going back to a basketball analogy, it's like you're really good in one place and then you go to another place, and grad school is like, going from undergrad to grad school is like, you're really good at your local park, whatever, and then you go to the NBA, and the game, the pace of the game in grad school is just faster. It's just, and it's different. In undergrad, you're studying for a test, and in grad school, you're studying to actually know something. And it's just like, you're not studying, grades are secondary in grad school because you need to be actually really good. And so like transforming your mind and thinking and what you consider to be success it's mentally taxing and it's hard. And so the things that you do, I think, is just, you keep pushing. The idea of celebrating the little victories got me through my first year. It's this idea of like, okay, didn't fail that. Like, and I mean little victories, like, okay, didn't fail that, like past that, like, okay, starting to connect the dots. And it's being patient with yourself. When you're applying for schools, the thing that I trusted my gut. When I went to Duke, it wasn't that they were trying to have me there as like a diversity thing. Like Duke really wanted me to succeed. And it became very clear when I was struggling my first 3 months or whatever, that, I mean, they put me in front of whoever I needed to talk through things, to get extra resources for stuff. Like they were not going to let me leave. Like that was very clear. Like, we're going to make sure you get out of here. And not every school is like that. And so asking the tough questions on these interviews, figuring out, and it's hard to see sometimes, but, ask other students, right? So I got this really good advice when I was going to grad school. Like, students are the most honest because they have no loyalty to the school, right? Like they're just there. And so if you really want to know what a university is like, what professors are like in terms of mentorship, ask, because they'll tell you like, oh, you don't want to work with so-and-so, like, or so-and-so favors this kind of student. So if you're not that, like, don't, it was very helpful for me when I went to Duke to talk to people like that. And so, it's hard to navigate, and I'm more than happy to help, even personal one-on-one, to really help people get through some of these things. So feel free to email me on this stuff because I'm really passionate about it. But it's something to think about a lot about at Brown, to put a plug in. It's not just about bringing students to Brown. It's making sure that students like excel at Brown. And not every institution has that secondary piece. They want to bring you there, but not necessarily help you excel and not help you navigate, not only the academic shocks of being in grad school and stuff is just faster, but also the cultural shocks sometimes if you're moving to certain places. And for me, like, it snows in certain places. I'm in California, like getting over like some of these things, right? And so I think, asking some of these hard questions and making sure that, it is a right fit. Don't go to a place because of the name. That's a huge thing. Go to a place that's going to maximize your ceiling. Those two places may not overlap. And that's a huge thing.
That's true. I mean, I went to UConn because they have really good basketball teams.
Duke let me shoot in Cameron Indoor, and I was like, oh yeah, I'm coming here. I think that was
I did my postdoc at Duke, so I was rivals. So you said I remember the qualifying exams, just for the students in the house, so to speak. Can you just talk a little bit, like, what qualifying exams are? Which ones did you take? And when you tanked on it, what did you do to not spiral into despair? Like, how did you keep going, right? What did you do in the face of failure?
Yeah, so I'll give the whole thing. I was on academic probation heading into my second semester. I took a class that gave me a little bit more confidence because I recognized that, like, maybe this may not come across via my talk because I've become very theoretical somehow, but I was not good at theory classes at first. I took my qualifying exam. I was on the border. And thankfully, actually, they failed me. And I appreciate that because it gave me a little bit more time. But I do remember when I failed thinking like, okay, this is it. I have one more chance. Qualifying exams for people are this kind of like this check to kind of make sure that everyone has some fundamental knowledge of the space. And a lot of times in statistics and math, they're written. So you have a written portion, which is, you have a set of problems that you need to kind of solve in some allotted time. And then you have an oral portion, which for statistics is not that bad because you're just presenting a research. For math, it can be traumatizing because I think it's like proof theorems orally and stuff. There's a lot of debate of whether or not these should stay, but the fact of the matter is they're around, and most schools give you 2 chances to pass them. I failed my first time, which means if I failed the second time, I would be effectively kicked out of the program. When I failed the first time, I remember talking to my advisor, who was just like, you just need more time. And he, again, going back to this idea of being around the sport a little longer, right? Like just getting used to, again, these intangibles of like the game is faster. I just need to sit in the material a little bit longer. So what I did my first year is that's when I joined Chris's group. And that was the best thing for me because I joined this biology lab where people knew nothing about statistics. So like I was a statistical expert in this group, and it gave me confidence. Like they came to me for stuff. And so I had to make sure I knew what I was talking about. And I got to play with data a little bit more without the pressures of grades. It just let me I got to code a little bit more without the pressure of grades. And it was nice. I got to just, get used to, have more of a feel for, what statistics was, certain concepts. And that was actually amazing. But it was support. Like I had people around me that were like, keep your head up kind of thing and keep pushing. And again, it's perseverance, right? You'll get it next time. And not everybody has that. And so again, this idea of being an inclusive environment was huge for me.
I think keep your head up. I think that was a song.
Yeah, seriously. I love it. It's tough, but yeah. You got to do it though.
Yeah, I failed one of mine and had that. Oh my God. I mean, analysis was like, analysis. Oh, okay. Love everything analysis. But the other, okay. Yeah. Well, but the great words of wisdom. So thank you, Dr. Crawford, for sharing your data science research work and also giving us insights on how to persevere and how to work through a myriad of data science opportunities. So Malik says, if you ever come to DC, we should play ball.
All right.
And perhaps maybe do a little bit of data. And you are always welcome to come back to Hotlanta. Well,
yes, as soon as it's safe to do so, I'm looking
I know,
forward
right?
To it.
Hopefully we'll get this all figured out. Sweet. But before I leave, somebody does have a question. We'll leave off with this question. What was your favorite CAU math class?
Oh man, there are 3 that come to mind. Differential equations with Dr. Rucker, I think was like a fundamental thing for me, which was I didn't want to do pure math. I wanted to do something that had a little bit more application stuff to it. Statistics with the Bebe was huge for me. And a kind of off the wall thing, which wasn't necessarily math, but I took a class with Dr. Minkins once, and that was insane. I still didn't appreciate like who he was like as a person, like in terms of how everyone else perceived him, and just like kind of learning from him. And I took a physics course with him was crazy. And so those are the 3 or 4 that kind of really stand out in my mind.
That's good. Well, I'm sure he'd appreciate that. So I talked with him earlier today, and Dr. Bebe has since retired. So I get to move into his office.
Yeah,
so
nice.
Super guy, super great guy. But again, thank you so much for giving us the keynote presentation. Great having you here. We're going to take a quick 15-minute break. People, y'all are welcome to hang out in the lounge and relax, put your feet up. We're going to check out the expo, but we'll be back here with the lightning talk starting at 1:15. So see y'all soon. Thanks again, Lorin.
Thanks for having me.